Okay. Okay. Very good afternoon, my dear friends. This is Dr. Padala Tirupati, Assistant Professor, Department of Botany. I'm in Government Degree College for Women, Jagityal. So today's our eminent resource person in front of us, dynamic personality, Dr.O.Padmaja Madam Garu, Assistant Professor, Department of Botany, Tara, Government College, Autonomous Sangareddy. Recently, college has got the great grade, that is A++. In entire Telangana, this is one of the best college in NAC grade because of our eminent resource person contribution is ever inspirable and appreciable. So, madam, please welcome and share your screen and start your session, madam. Please welcome.
Okay, thank you, Tirupati sir. Thank you very much, and all the credit goes to you also, sir, for performing this good coachings for all the students. I'm going to start my session. So, good afternoon, students. Myself, Dr. Padmaja, Assistant Professor of Botany, Tara Degree College, Sangareddy. Today, we will discuss about the remaining part of respiration, very important topics. And we can expect some bits from this chapter also. So the continuation of our previous class. In previous class, we discussed regarding the glycolysis, and the end product of glycolysis is pyruvic acid. The pyruvic acid is a 3-carbon compound. And now, this pyruvic acid now entering into the after glycolysis, the next is the Krebs cycle. So the end product of glycolysis is pyruvic acid. It is a 3-carbon compound, and the oxidation of pyruvic acid into CO2 and water in mitochondria is called as the Krebs cycle. The Krebs cycle is taking place in mitochondria, and it was discovered by Krebs. So, in 1936, by its name, the cycle name is a Krebs cycle, and this is also called as citric acid cycle, because the first common compound from the pyruvic acid is citric acid. So it is called as citric acid cycle. And it is also known as tricarboxylic acid cycle. That means 3 carboxylic acid containing groups are there. So it is also called as tricarboxylic acid group. Since the first product, citric acid, has 3 carboxylic group. And this pathway is also known as mitochondrial respiration or oxidation of pyruvate. And this Krebs cycle is an aerobic process. It occurs in the matrix of mitochondria. This Krebs cycle involves the following steps, because if they may ask the cycle, regarding the cycle also they may ask, or depending upon the how many ATPs are formed by the end of this glycolysis or Krebs cycle or electron transport, like this, they may ask the questions from this point of. And now here, of acetyl choline. This acetyl CoA is a 2-carbon compound. The pyruvic acid is a 3-carbon compound. So here what happens? This pyruvic acid 3-carbon compound will not enter into this mitochondrial map. So it must be converted into acetyl CoA. And now this pyruvic acid combines with CoA to form acetyl CoA. And this acetyl CoA is a 2-carbon compound. Pyruvic acid is a 3-carbon compound. One CO2 is released from this process. So in this reaction, 2 hydrogen atoms and 1 CO2 molecules are removed. Hence, this reaction is called as oxidative decarboxylation. As a result of this CO2 removed from this pyruvic acid, it becomes a 2-carbon compound that is acetyl CoA. This reaction is catalyzed by enzyme pyruvic acid dehydrogenase. So every step must be catalyzed by the enzymes. Now the enzyme that is participated in order to convert this pyruvic acid to acetyl CoA. So if you see the equation, the 2 hydrogen atoms are accepted by NAD to form NADH2. So it is accepted to form NADH2, and pyruvic acid is converted into acetyl CoA and CO2 carbon dioxide released. So your decarboxylation is taking place, and in presence of by taking this NAD and giving rise to NADH2, and 2 hydrogen atoms are also participating. So oxidative decarboxylation. It combines with oxaloacetic acid to produce 6-carbon compound, that is citric acid. And the acetyl CoA is a 2-carbon compound, and the oxaloacetic acid is a 4-carbon compound that is formed by the process of the citric acid to this acetyl oxaloacetic acid. This citric acid is a 6-carbon compound. So here the first formed compound is a citric acid. So the cycle is also called as citric acid cycle. This reaction is catalyzed by the enzyme citric acid synthetase with the release of CoA. Now what happens? This acetyl CoA combines with oxaloacetic acid. This acetyl CoA 2-carbon compound and oxaloacetic acid 4-carbon compound. 4-carbon compound, and the COA is coming, and as a result, this is converted into the citric acid. And the next step is the dehydration. Now the citric acid undergoes dehydration in the presence of enzyme ecolipase to form cis-ecolic acid. This citric acid, in presence of enzyme dehydrogenase, is converted cis-ecolic acid and releasing the water. So it is called dehydration step. And next one is the hydration. This we have to remember that by which substance is converted to this substance in presence of which enzyme, that we have to remember. And now this formed compound is a cis-ecolic acid. Now cis-ecolic acid undergo hydration with the help of an enzyme ecolipase to form isocitric acid. That means in presence of water, it is converting into this cis-ecolic acid is converting to isocitric acid. And the next step is the dehydrogenation and decarboxylation step. This is an important step we have to remember because this ketobutyric acid is coming intermediate in different pathways. This isocitric acid is dehydrogenated and decarboxylated to alpha ketobutyric acid and CO2 by isocitric acid dehydrogenase. Here what happens? Here isocitric acid is a 6-carbon compound. It is converting to 5-carbon compound, that is beta-glutaric acid, by releasing the CO2. And NAD is taking hydrogens and giving rise to NADH2. That means in every step the CO2 or NADH2 formation or water, the removal of water is taking place. And the next step is the oxidative decarboxylation. This alpha ketoglutaric acid undergoes oxidative decarboxylation to form succinyl CoA by the addition of CoA. Now, this reaction is catalyzed by alpha ketoglutaric acid dehydrogenase with the help of NAD. So here what happens that ketoglutaric acid and CoA NAD is in presence of an enzyme alpha ketoglutaric acid dehydrogenase, giving rise to succinyl CoA and NADH2 plus the CO2 will be coming outside. Now, the succinyl CoA is a 4-carbon compound. Alpha ketoglutaric acid is a 5-carbon compound. By removing another CO2, this 5-carbon compound is converting to 4-carbon compound. They may ask, how many carbon compound is alpha ketoglutaric acid, or how many carbon compound is succinyl coenzyme, like this, or which step it is? It is the oxidative decarboxylation step, or by which step this ketoglutaric acid is converting to succinyl coenzyme. Like this, they may ask the gifts in this. And next, this is the overall skeleton of a citric acid cycle. Now, what happens? This pyruvic acid compound giving rise to acetyl CoA. This acetyl CoA combines with oxaloacetic acid to give rise to citric. So this cycle is also called the first formed compound is citric acid. So it is also called a citric acid cycle. And here oxaloacetic acid is a 4-carbon compound, acetyl CoA is a 2-carbon compound, citric acid is a 6-carbon compound. Now this citric is converting to cis aconitate, and cis aconitase is converting to isocitrate. And here the NAD is giving rise to NADH2, and isocitrate is converted to oxalosuccinate, and by releasing CO2. As a result, this oxalosuccinate is a 6-carbon compound. By removing one CO2, it is forming a 5-carbon compound, that is 5-ketaglutarate. This alpha ketaglutarate is converted into succinyl CoA. Succinyl CoA is a 4-carbon compound. Now again the CO2 is released and NAD is taking H plus and giving rise to NADH2. Whatever we are talking about this NADH2 and this the GTP, this will have to remember that for the balance sheet of the respiration. Now, succinyl CoA taking succinyl CoA is again giving rise to the GTP that give by taking the GDP plus phosphate give rise to succinate. Again, this succinate, it is a 4-carbon compound. It is converting to fumarate, and this FAD is giving rise to FADH2. And fumarate, by giving the water molecules, is converting to malate, and this malate again converting into oxaloacetate. This oxaloacetic acid is a 4-carbon compound. Now it is giving rise to NAD is taking H2H plus to give rise to NADH2. So what we have to mainly remember this in citric acid cycle is what are the carbons compounds are there, and the releasing of NADH2, the formation of NADH2, and where the CO2 is released, and how many carbon compound is each one. Or they may ask what are the enzymes, what are the steps are essential to converting one substrate to another substrate. And acetyl CoA is hydrolyzed to succinic acid by enzyme succinyl thiokinase. Here CO2 is released, ADP is phosphorate, inorganic phosphate to form ATP, In animal cells, GDP reacts with inorganic phosphate to form GTP. That means that ATP or GTP is the same. So succinyl-CoA is converting to succinic acid by releasing CoA, and ADP is combining with the phosphate to give rise to ATP. And next is the oxidation. The succinic acid is oxidized to fumaric acid. The succinic acid is also a 4-carbon compound. Fumaric acid is also a 4-carbon compound. This reaction is catalyzed by succinic acid dehydrogenase with FAD. Now the FAD takes H plus ions to give rise to FADH2. This flavin adenine dinucleotide. That is the full form of FAD. Now, succinic acid combining with FAD in presence of an enzyme, succinic acid dehydrogenase, it is converting into fumaric acid and FADH2. Now, the fumaric acid reacts with water to form malic acid. This reaction is catalyzed by the enzyme fumarase. So, by the equation, the fumaric acid combines with the water in presence of fumarase to give rise to malic acid or malate. And dehydrogenation. Malic acid is converting to oxaloacetic acid. The first starting of this Krebs cycle, we told that this oxaloacetic acid combines with the acetyl-CoA. So this oxaloacetic acid, by the series of reaction, the oxaloacetic acid is formed, and by the enzyme malic dehydrogenase with the help of NAD. Oxaloacetic acid then accepts another molecule of acetyl-CoA to continue the next round of Krebs cycle. Like this, the continuation will be taking place. Now, this malic acid is converting to oxaloacetic acid, and NAD is converting to NADH2. This is the steps that are taking place in the Krebs cycle. It is mainly taking place in the mitochondrial matrix, and it was also called a citric acid cycle. And the Krebs discovered this, so the cycle is also called as Krebs cycle. And this Krebs cycle is also called as amphibolic pathway. In the next slide, you can learn that one. This is also a bit weaker. And next is the ETS, that means electron transport system. So, by all the process that must be converted into ATP, the energy currency. So, how they are moving from one electron carrier from complex 1, 2, 3, and how the ATP formation will be taking place, that may be asked in the bits. So, that electron transport system is also called as oxidative phosphorylation. In photosynthesis, the formation of ATP is called as photophosphorylation. In respiration, it is called as oxidative phosphorylation. And what is the diagram that is represented, that is seen in the membrane of the mitochondria. So, the electron transport system is the metabolic pathway present in the inner mitochondrial membrane. Just for our simply study, we are draw this diagram, but actually the same will be present on the membrane of the mitochondria. You see there inner membrane space and inner mitochondrial membrane, and here it is the matrix. So, before starting our lesson, we will also discuss the structure of mitochondria. This mitochondria is the powerhouse of the cell where the respiration is taking place. And here the electron transporter. Here the inner mitochondrial membrane is also called as the crystine. That means this membrane is going into number of finger-like projections. They are called as the crystine. And here this electron transport chain is taking place on the crystine of the mitochondria, and the inner membrane space, the space between the outer mitochondrial membrane and inner mitochondrial membrane is called as intermembrane space. And now here This electron transport system is the metabolic pathway present in the inner mitochondrial membrane through which the electron passes from one carrier to another carrier. So the moving of these electrons from one carrier to another carrier, and this is to release and utilize energy stored in the NADH2 and FADH2. These are the products that are formed during glycolysis and the Krebs cycle. They are NADH2, FADH2. By oxidation, the electrons are passed onto the oxygen to form water. And now electrons from NADH are oxidized by an NADH dehydrogenase. Here, for our easy study, we marked as complex 1, complex 2, complex 3, and complex 4 and complex 5 as ATP synthase. That we have to remember that ATP synthase or the coupling factor. This complex 5 is considered as an ATP synthase. These are all arranged on the inner mitochondrial membrane. And here, electrons from NADH are oxidized by NADH dehydrogenase, that is complex 1. So complex 1 is known as NADH dehydrogenase, and electrons are then transformed to ubiquinone Q located within the inner membrane space. That means from complex 1 to complex 2, the moving of the electrons are taking place by the electron carriers. So the electrons are then transferred to ubiquinone located within the inner membrane. Ubiquinone also receives reducing equivalents via FADH2, complex 2. So the complex 2 is FADH2 that is generated during the oxidation of succinate in citric acid cycle. And the reduced ubiquinone is then oxidized with the transfer of electrons to cytochrome C via cytochrome BC1 complex. That means complex 3. The complex 3 is also called as cytochrome BC1. The cytochrome C is a small protein attached to the outer surface of the inner membrane. Cytochrome C, it acts as a mobile carrier. They may be asked that which is a mobile carrier for the transport of electrons between complex 3 and complex 4. That is the cytochrome C. In the diagram also, you can see the cytochrome C that is moving from one place to another place. These are the fixed positions. And now the complex 4 is refers to cytochrome C oxidase. Complex containing cytochromes A and A3 and 2 the copper centers. So this from one electron complex to another, the movement of electrons are taking place. Just we have to mainly remember that the complex 4 is cytochrome C oxidase complex. And now the important one is here that complex 1 is NADH dehydrogenase, complex 2 FADH2, complex 3 cytochrome BC1, complex 4 cytochrome C oxidase, and complex 5 ATP synthase. This ATP synthase is an important one. When the electrons pass from one carrier to another via complex 1 to 4, they are coupled to form ATP synthase. So how the ATP is forming and what is the role of this ATP synthase or complex 5? And for this is mainly for the production of ATP. So the diagram, if you see the diagram there, the complex 1, 2, 3, and 4, and the fifth one is this ATP synthase. This is the coupling of ADP plus PI and ATP formation we are seeing. And the important bit you have to remember is number of ATP molecules produced depend on the nature of the electron donor is 1 NADH fluoride is equivalent to 3 ATP molecules. Thus we have to remember what that was. and FAD is equal to 2 ATP molecules. So the oxidation of one NADH is containing 3 ATP molecules energy, and oxidation of one FADH2 contains 2 ATP. That we have to remember. And the complex 5 as a ATP synthase. This is also called as the coupling factor. Why it is called as the coupling factor? Means it is combining ADP plus PI to form ATP. So it is also called as coupling factor or ATP synthase. Now, this ATP synthase and chemiosmosis process is told by the Mischel. According to Mischel, what is the main theme of this is if you see the diagram, in inner membrane space, more electrons are accumulated. So the concentration gradient of protons are there in the inner membrane space. So these concentration gradient helpful for the moving of protons from inner membrane to the inside. As the moving of these, the coupling, these concentration gradient helpful for the coupling of ADP plus PA to form the ATP. So see in the lower diagram, if we see that the moving of ATP molecules as the concentration gradient is more in the inner mitochondrial space, when they are moving, this gradient helpful for coupling this ADP plus PA to form ATP. This process is taken by the ATP synthase. So the energy released during electron transport is utilized to synthesis ATP by ATP synthase complex. And this ATP synthase consisting of two major components. That one is the F1 and FO. F1 is the headpiece, that is the peripheral membrane protein complex. F0 is the integral membrane protein complex. This F1 and F2 is called as the ATP synthase or the coupling factor. And F1 headpiece, site of ATP synthesis from ADP and inorganic phosphate. So this ADP and PI, that means inorganic phosphate, combining to form a ATP. And it forms a channel through which protons cross the inner membrane. So these in the inner mitochondrial space, more amount of H+ ions are there. The concentration is more. In order to move from inner mitochondrial space to inside the mitochondria membrane, this is permeable. Only this ATP synthase is permeable to move the H+ ions. So the H+ ions only can move through this ATP synthase. While they are moving, this proton gradient helpful for coupling of this ATP plus PI to form the ATP. And actual, this is the diagram representing in the mitochondria. So this is a mitochondria, outer mitochondrial membrane. You all know that every membrane is made up of lipids and proteins. And in between, the space between the outer membrane and inner membrane is called as the inner membrane space or perimitochondrial space. And here these are the complex 1, 3, 2, 4, and 5. Like this, they are for our easy study. We are considered as a complex 1, 2, 3, 4, 5. These are present on the inner mitochondrial membrane. And here the liquid substance that is present in the mitochondria is called as the matrix. And here this ADP oxidation and the reduction of this ADP, FAD as taking place, more amount of H+ ions are accumulated in the inner membrane space. This electron concentration gradient helpful for making a movement of this ATP synthase for coupling of this ADP plus PI to give rise to ATP. If you see the diagram also, here the ADP plus PI giving rise to ATP. So this coupling force is coming from this concentration gradient of H plus ion. And if you see for our easy understanding, in the glycolysis, the end product is pyruvate, and the pyruvic acid 3 carbon compound is not directly participating in the Krebs cycle. It must be converted into acetyl-CoA. So it is considered as the link between the glycolysis and Krebs cycle. And next, the acetyl-CoA is combining with oxaloacetic acid entering into the Krebs cycle. So first formed compound is the citric acid, and now this Krebs cycle, when now they are entering into the electron transport, this electron transport is helpful for the formation of the ATP molecules. So net gain of ATP from each glucose molecule is calculated based on the following assumptions. Now, all the steps in glycolysis or TCA cycle or electron transport occur sequentially and orderly. We already studied that 1 NAD is equal to 3 ATP molecules. The NAD is synthesized in glycolysis, is transferred into mitochondria and undergoes oxidative phosphorylation. Intermediates in the pathway are not used to synthesize other products. Only glucose is being respired. Other alternative substrates are not entered in the pathway at any stage. And now, this is we have to remember this, a very important one. Total, the glucose is converting into pyruvic acid. In this, the ATP molecules, how many ATP molecules are formed in the ATP in the glycolysis? So 2 ATP directly gives 2 ATP molecules, 2 molecules of NADH. That 1 NADH is equal to 3 ATP molecules. So 2 NADH is equal to 6 ATP molecules. And oxidative decarboxylation. 2 NADH is equivalent to 6 ATP molecules. In TCA cycle, that means Krebs cycle, 6 NADH are formed, 2 FADH are formed, and 2 GTP are formed. That means one pyruvic acid is participating in this, another pyruvic acid is participating in this Krebs cycle. So 6 3s are 18. And FAD is equal to 2 ATP molecules, 2 2s are 4, and GTP is equivalent to 1 ATP. So 2 ATP, the total is 13 ATP formed. So 2 ATP molecules are spent for transporting 2 NADH molecules formed during glycolysis. So hence the net gain is 36 ATP molecules. So they may be asked the question regarding this, or they may be asked that 1 NADH is equal to how many ATP molecules. And next is the comparison between the ATP formation between the fermentation and the aerobic respiration. We already know that the fermentation comes under the anaerobic respiration. The anaerobic respiration is nothing but the partial breakdown of glucose, and the net gain of only 2 ATP molecules are there. And here completely breakdown of glucose will be taking place, and net gain is the 36 ATP molecules. And now, this is some maybe ask this, why this citric acid cycle is called as an amphibolic pathway. See here, these are the substrate, the fats, the carbohydrates, and the proteins. So end products of the fats is the fatty acid and glycerol. And carbohydrates, these carbohydrates may be the separate pathways will be there for the fats and the proteins also. But in our study in the respiration, only we are discussing about the carbohydrates. That means the glucose of the sugar. The end product is by the photosynthesis. The glucose is converted into glucose 6-phosphate, fructose 1,6-phosphate. And here the fructose is bifurcates into dihydroxy S1 phosphate and dehydrated glucose. That means that maybe this substance will become an intermediate in another pathway. For this process, the bifurcation of this fructose 1,6 biphosphate is converted into dihydroxyl phosphate and glyceraldehyde 3-phosphate. That glyceraldehyde 3-phosphate at the end gives rise to pyruvic acid. This pyruvic acid, by converting the acetyl-CoA, converting the entering into the Krebs cycle, and from there, this electron transport system is helpful for the releasing of the energy currency ATP molecules. And the end products of the proteins are the amino acids, and at last the amino acids come into pyruvic acid. Again, this the same cycle will be continued here. And now the respiration pathway is generally considered as the catabolic pathway. That means the breaking of these food substrates are taking place in presence of oxygen. That means the complex molecules is giving rise to simple molecules. So this pathway is called, this metabolic system is called as the catabolic pathway. This respiration is called as catabolic pathway. Respiration is called as anabolic pathway. This big complex is splitting into giving rise to small molecules like carbon dioxide, water, and some amount of energy is released. So this respiration is considered as a catabolic pathway, and it involves both the anabolism and the catabolism. The synthesis will be taking place and the breakdown will be also taking place. So it is better, it is called as a amphibolic pathway. So example, fatty acid breakdown to acetyl-CoA before entering the respiratory pathway. But when the organism needs to synthesize fatty acid acetyl-CoA, withdraw from the respiratory pathway. Similarly, during breakdown and synthesis of protein, respiratory intermediates are involved here. So it is the intermediate. So number of intermediate products are formed in this Krebs cycle. So it is also called as the amphibolic pathway. The Krebs cycle is called as amphibolic pathway, and this also we studied, but is again important after completing this respiration. RQ is the formula is volume of CO2 evolved by volume of oxygen consumed. So this down equation, if you see the photosynthesis, respiration, 6 C6H12O6 in presence of oxygen, giving us 6 molecules of CO2 and 6 molecules of water. So what is the formula for RQ? Volume of CO2 evolved by volume of oxygen consumed. So how many here CO2 is 6 and 6 oxygen. 6 divided. So it gives to the 1. So RQ of carbohydrates is 1 and RQ of fats is less than 1 and RQ of proteins is equal to 0.1. Okay, and now just you have to remember this equation and the RQ values. So this is the end of chapter respiration. The mainly they will be focused on the glycolysis. In the glycolysis, what is the end products and where the glycolysis is taking place in the cell. That means cytosol or the cytoplasm, and where the Krebs cycle is taking place, and mainly they will ask that ATP synthase, the coupling factor, or oxidative phosphorylation. So this due to this concentration gradient of H+ ions helpful for the formation of these ATP molecules. Okay. Thank you.
Madam.
Sir.
Okay, any doubt regarding of this class, my dear friends? I hope Madam has clearly explained about the TCA cycle or Krebs cycle or amphibolic pathway or citric acid cycle, one of the most important cycle in respiration, and also in plant physiology. This cycle, it is useful for both metabolic activities like catabolic and anabolic processes. So Madam has clearly explained all the stages. So all the phases of this cycle, total 10 stages, and also the respiratory quotient and the balance sheet and the ATPs how many ATPs are involved in this process. So Madam has clearly explained about even though enzymes, each stage, which type of enzymes are involved in this process. So really wonderful class, Madam. Thank you very much. So tomorrow we will meet in same time. Thank you, Madam.
Thank you, sir. Thank you.
Right.